previous lecture, we have already started, we had studied the chromatin organization. Chromatin organization, that is the packaging of DNA inside the eukaryotic chromosome. So in the last lecture or in the previous lecture, we had a preliminary discussion on the chromatin organization, and today we have already we have to discuss the structural organization of the eukaryotic chromosome. You can say that, you can also say that this is the chromatin organization, how the DNA is organized in the form of chromatin structure. And this topic is also known as nucleosome model. So now let us look at the whiteboard and try to understand the diagrams here. Look at the whiteboard. Let me know whether the whiteboard is visible to you. Hello? Hello? Hello?
Yes, sir.
Whiteboard दिख रहा है?
Yes, sir.
देखो
सर शाइन कर रहा है.
थोड़ा सा साइन कर रहा होगा. देखिए फिर से हम ये तो हम शेयर कर ही दिए हैं, व्हाट्सएप ग्रुप में शेयर किया हुआ है ना फोटो डायग्राम. हम तो सिर्फ समझाने के लिए दिखला रहे हैं. आप देखिए यहाँ DNA. Just I have written just let me explain this the level of coiling of the DNA to form the nucleosome structure, solenoid structure, super solenoid and chromosome. देखो, just I have drawn a flow sheet on the whiteboard. You all know that the length of the DNA is much more than the length of chromosome, and it is also very clear to you that the diameter of the DNA, or you can say that the thickness of the DNA or diameter of the DNA is 2 nm. 2 nm, and this 2 nm fiber of the DNA undergoes first level of coiling to form a number of repeating units, and these repeating units are known as the nucleosome subunits. And this first level of coiling, when the 2 nm fiber of the DNA undergoes coiling around the histone protein to form nucleosome subunits, and these nucleosome subunits are the nucleosome subunits are formed due to the chemical interaction between the histone protein and the DNA. And this and the diameter of this nucleosome is the 10 nm or 11 nm, and this structure is formed as a first level of coiling of DNA around the histone protein. And you all know that histone protein is a basic protein because of the presence of basic amino acid. And this nucleosome structure, which has a diameter of 10 or 11 nm, again undergoes second level of coiling to form a structure which is known as the solenoid. And now due to the second level of coiling of the nucleosome to form the solenoid structure leads to the formation of the solenoid structure, and the diameter of this solenoid structure is 30 nm or 300 angstrom, and this structure is now called coil of the coil. And this solenoid structure again undergoes for third level of coiling to form super solenoid structure, and the diameter of this super solenoid structure is the 400 nm or 4000 angstrom. And this is this solenoid structure undergoes coiling to form super solenoid structure, and this level of coiling is known as the third level of coiling. And you can also say that this is the super coiling. And this super solenoid structure again undergoes further coiling, which you can say that the final level of coiling to form chromatid or chromosome. So this is the level of coiling. You know that the length of the DNA is much more than the length of the chromosome. And for the packaging of DNA inside the chromosome, the coiling of the DNA, the coiling of the DNA around the histone protein to form the nucleosome take place, and this nucleosome structure undergoes further coiling to form the solenoid structure, and this solenoid structure undergoes the third level of coiling to form super solenoid structure, and this super solenoid structure in turn undergoes final level of coiling to form chromatid and chromosome. So the first level of coiling leads to the formation of nucleosome. The second level of coiling leads to the formation of solenoid. The third level of coiling leads to the formation of supersolenoid, and the final level of coiling leads to the formation of chromatid and chromosome. And as we as the level of coiling increases from the first step to the final step, the diameter of the chromatin thread increases. As you know that the DNA is a very thin structure, thin, and its diameter is 2 nm, and the diameter of nucleosome is 10 nm. The diameter of solenoid is 20 to 30 nm. The diameter of supersolenoid structure is 400 nm, and finally due to the final compaction, there is this final compaction results in the formation of the chromatin or a chromosome, chromatid or chromosome. Now look at the whiteboard here and try to understand these structures. These structures drawn on the whiteboard. Look here. This is the one nucleosome subunit. This is the histone. The green circle drawn on the whiteboard shows histone protein. This is the one histone protein. This is the second histone protein. This is the third one. This is the fourth, fifth, and so on. So a particular histone protein which is around the histone protein, the DNA is wrapped. The DNA is wrapped, and this is the structure of one nucleosome subunit. Look here. This is the structure of one nucleosome subunit. In this structure, we have shown two molecules H2A, two molecules of H2B, two molecules of H3, and two molecules of H4. So there are two molecules of H2A, H2B, two molecules H3, and two molecules H4. So in totality, there are 8 histone proteins consisting of H2A, H2B, H3, and H4, and two molecules of each histone protein. And these histone proteins form the central core. Around this central core, around this central core, the DNA, the length of DNA is wrapped, and the wrapping of the DNA around the histone protein makes 1.75 turn, and the length of the part of the DNA which remains outside the nucleosome subunit is known as the linker DNA or a spacer DNA, with which the another histone protein which is called H1 protein is attached. This is the H1 protein remains attached. So, and this is the formula of nucleosome. Nucleosome is equal to 144 base pair plus 2 molecule each of H2A, H2B, H3, and H4 of DNA. And nucleosome is just defined as the nucleosome acts as a fundamental subunits of the chromatin thread, and the solenoid is the coil of the coil. So this flow sheet diagram shows the level of coiling of the DNA for the packaging of DNA inside the chromosome. And again, I just want to say that this nucleosome structure, the term nucleosome was coined by P. Audet in 1975, and this nucleosome structure was for the first time reported and discovered by R.D. Korenberg in 1974. And this nucleosome structure, what is nucleosome? Nucleosome is defined as the interacting complex of the interacting complex of DNA with the histone protein, and as a result of which a string of bead-like structure appears, and these bead-like structures are known as the nucleosome subunits. And the DNA at first undergoes first level of coiling to form nucleosome. The nucleosome undergoes second level of coiling to form solenoid. The solenoid undergoes third level of coiling to form supersolenoid. And finally, the supersolenoid structure undergoes final coiling to form the chromatid and chromosome. So this is all about the diagram and the way in which the formation or packaging of DNA inside the eukaryotic chromosome takes place. Am I clear to all of you? Yes,
sir. Yes.
Again, I want to mention here the main points. Be sincere and try to understand and try to listen the main points again. Nucleosome. What is nucleosome? Nucleosome is the interacting complex of the DNA with the histone protein. The DNA, a particular length of the DNA, chemically interact with the histone protein to form the bead-like structure, and each bead-like structure is known as the nucleosome subunits. And there are so many nucleosome subunits, and as a result of formation of a number of nucleosome subunits, the chromatin appears like a string of bead-like structure, and these beads represent the nucleosome. And you know that for the interaction of the interaction of DNA with the histone protein provides the best example of nucleoprotein interaction. You know that the nucleic acid interact with the protein. So this is also a very good example of the interaction between the protein and nucleic acid. The formation of nucleosome can be cited as the best example of nucleoprotein interaction, because in this case the nucleic acid DNA is interacting chemically with the histone protein. So this is also a very excellent example of the nucleoprotein interaction. You have also studied the protein carbohydrate interaction, when you might have studied the plasma membrane oligosaccharide molecules are found there. Similarly, here this in case of nucleosome, nucleic acid is chemically interacting with the protein. As you know, the nucleic acid is acidic in nature and histone proteins are basic in nature. Therefore, this association of nucleic acid with the protein also helps in the neutralization of the acidic nature of the DNA. So this is the first very important function of the nucleoprotein interaction. And the second and the very important why this structure is formed. This structure, nucleosome, is formed for the packaging of DNA inside the eukaryotic chromosome. And this nucleosome, the concept of this nucleosome came into existence due to the discovery by R.D. Korenberg in 1974, and these repeating units were known as the nucleosome, and the term nucleosome was coined by the P. Audit in 1975. So this DNA, which is 2 nm in thickness, undergoes first level of coiling around the histone protein to form a 10 nm structure, bead-like structure, and these 10 nm bead-like structures are known